So yesterday in last class, we studied about the what is carbohydrate. Now, tell me what do you understand by carbohydrate? Function part you read from the book. It's not that tough. In properties of monosaccharide, isomerism is asked as short note. Okay, isomerism, stereoisomerism, then anomerism. These are the things they ask. So, roll number 80, 90. What is anomerism?
What is?
Anomerism. Yes. Okay, isomerism is same formula but different spatial arrangement of molecules. Okay, different structure you say. So that is the isomerism. But there is another thing is anomerism. Anomerism. They will ask you what is anomerism, or in Viva you will be asked what is anomeric carbon. Okay. So we told that there are two functional groups, aldehyde group or keto group. Now, in case of two monosaccharide, one of the monosaccharide hydroxyl group of the anomeric carbon keto group reacts with hydroxyl group of another monosaccharide may be of the aldehyde or keto group or any other hydroxyl group. Okay. In that case, the carbon of, this is called carbonyl carbon, carbon atom of aldehyde or keto group, that becomes an anomeric carbon. Okay. Asymmetric carbon because of attachment of different four molecules. Okay. Now, in this case, where this carbon of hydroxyl group, this one aldehyde group or keto group becomes anomeric asymmetric carbon, in that case this asymmetric carbon is called anomeric carbon. Okay. So, just try to know what is anomeric carbon. Isomerism, stereoisomerism, epimerism. Okay. Epimerism, what is that? Epimerism, while two isomers differ in configuration in the respect of one carbon only. Okay. Glucose and galactose, they differ in spatial arrangement of hydroxyl group in carbon number 4 only. So they are, galactose is epimer of glucose. Okay. Mannose is again epimer of glucose in respect of carbon number 2. Okay. So these are the epimerism. So we will proceed to our today's topics. Disaccharide. Another thing I forgot to tell you, look up this Mutarotation also. When this in anomeric, when there is anomeric carbon is present in the in some monosaccharide, there is another isomer used to be formed, alpha and beta. Okay, alpha and beta form, alpha form and beta form. So in glucose, alpha D glucose and beta D glucose, these are the anomers, and they have specific rotation. In case of alpha D glucose 112, in case of beta D glucose it is plus 18. So when it is in mixture, there is exchange of this alpha and beta glucose, and it comes to an equilibrium, and that time the specific rotation it becomes 52 plus 52.7. Okay, and this change in specific rotation of light, polarized light, is called mutarotation. Okay. So the change in the specific optical rotation representing interconversion of an alpha and beta form of the glucose to an equilibrium mixture is called mutarotation. That also sometimes is given in short note. See, it may have 5 numbers, mark carrying 5. You don't have to write this much, okay? Just write this thing, you will be given mark. Disaccharide. So, as you have told, there will be 2 monosaccharide bound by glycosidic bond and form a disaccharide molecule. These 2 monosaccharides may be the same monosaccharide or may be different monosaccharide. Okay. So, it consists of 2 monosaccharide unit held by glycosidic bond. They are crystalline, water soluble and sweet to taste. Okay. Now, there are 2 types depending of capability to reduce cupric oxide in alkaline medium. They are divided into 2, reducing disaccharide and non-reducing disaccharide. Reducing disaccharide is maltose and lactose, and non-reducing is sucrose. Sucrose means table sugar, the cane sugar what we take in every day. This is sucrose. This is non-reducing. Remember, sucrose is non-reducing. In Viva, in practical, everywhere you will be asked. Okay? And reducing is maltose and lactose. Maltose, it has 2 glucose units bound by alpha 1,4 glycosidic linkage. Alpha 1,4. Just remember, alpha 1,4 glycosidic linkage 1 and 4. In case of first glucose molecule, it will be first carbon. That first carbon carries what? Aldehyde group. And in case of second glucose molecule, it will be fourth carbon, which is not a aldehyde group. Okay. This is the hydroxyl group of the carbon which doesn't belong to aldehyde group. So one is having carbon number 1, which was aldehyde group, and another is the carbon number 4, which is not aldehyde group. So second carbon, the aldehyde group of first carbon is free, no? It is free. That's why in maltose it is reducing. It is reducing because in second glucose molecule, the first carbon, which is an aldehyde group, is free to react with copper cupric ion. So it is one of the reducing disaccharide which has free functional group. This is the structure. Structure you refer for your understanding, you may not write in examination. So this is the first carbon, 1, 2, 3, 4, 5, 6. In case of solution, the monosaccharide spontaneously undergoes cyclization and forms this pyranose, glucopyranose form of structure. There is 6-membered cyclic monosaccharide. Okay, so this is the 1. This is carbon number 1. 1, 2, 3, 4. So it has a glycosidic bond between carbon number 1 of glucose number 1 and carbon number 4 of glucose number 2. So this C, this is free. This carbon is free, having aldehyde group, and it is responsible for reducing property of maltose. Lactose, it is linked by beta 1,4 glycosidic linkage. Monosaccharide units are galactose and glucose. Okay. Enzyme responsible for hydrolyzing the lactose is lactase or beta D galactosidase. The source of carbohydrate is breastfed infant. In breast milk, it is there. Lactose, as you know, milk is the source of this lactose. So here, beta, because where it was downwards, it was alpha. But in this case, the hydroxyl group is upwards. Okay. Now, in comparison to this, this is towards the group, alcohol group. So, this is beta D galactose, and here it is beta D glucose. Okay. So, glycosidic bond is beta 1,4 glycosidic bond. Now, the sucrose linked by alpha 1,2 glycosidic linkage, it is non-reducing disaccharide. Why non-reducing? See, this is the glucose, this is the fructose, this is 6-member cyclic form, this is the carbon number 1, this is the 5-membered cyclic form, this is carbon number 2. It starts from 1, 2, 3, 4, 5, 6, and here it starts 1, 2, 3, 4, 5, 6, 3, 4, 5. Okay. So, the first carbon of glucose, which is anomeric carbon and forms a glycosidic bond with the second carbon of fructose, which is again an anomeric carbon. So, both the anomeric carbons has taken part in glycosidic bond formation. That's why it doesn't have any other carbon which is having keto group or aldehyde group free for reducing property. That's why this sucrose is non-reducing. So, remember, sucrose is non-reducing. Isomaltose, it is also contains 2 moles of glucose. They link by 1,6 alpha 1,6 glycosidic linkage. Now, when in case of say polysaccharide glycogen, we go on hydrolyzing the this molecule, glycogen molecule. It is bound by 1,4 linkage, but at the branch it will have 1,6 linkage. So, it will release one molecule of, or when one branch is hydrolyzed, so it will ultimately release one molecule of isomaltose. Okay. So, it is like a maltose, but bond is different, that's why it is called isomaltose. Okay. Now, the enzyme which hydrolyzes alpha 1,4 glycosidic bond is not capable of hydrolyzing alpha 1,6 bond. Okay. That's why it is separate. This disaccharide is separate, and the isomaltase is the enzyme which can hydrolyze this bond. Okay. Trehalose, another disaccharide, alpha 1,1 glycosidic linkage, and trehalase is the enzyme responsible for hydrolysis, and it is found in yeast and fungi. So, coming to oligosaccharide, 3 to 10 monosaccharides they will have, because I have separated disaccharide as separate group. So, oligosaccharide, they join together by a specific bond called glycosidic bond. On hydrolysis, they give 3 to 10 molecules of simple sugar. They are subdivided based on the number of monosaccharide. These are trisaccharide, tetrasaccharide, pentasaccharide. Okay. So, this is the pentasaccharide. It will contain 3 galactose, 1 glucose and 1 fructose. They are mainly responsible for glycosylation of the protein. Coming to the polysaccharide, there is repeat unit of monosaccharide or their derivatives held together by glycosidic bond. Their function is storage function or structural function. Example, starch in plants and glycogen in animal. They contains more than 10 sugar units. Their molecular weight is very high. They are also called glycans. They may be either linear or branch structure. Branch structure or linear structure or branch structure. Suppose this is a sugar unit. This is sugar unit. Monosaccharide, I mean. This is branch. So this bond will be 1,4 alpha 1,4. This will be 1,4. This is 1,4, but this will be 1 and 6. This branch site will be 1 and 6. Again, this will be 1,4. This is 1,4. Okay. So like that, it appears like a tree. It will appear like a tree. But it is unlike that of protein, which is not branched, or nucleic acid. This is also not branched. They will go on in protein. There will be sequential attachment of amino acids in a linear chain. There may be folding. They may fold, undergo folding of 2-3 times, but there is no branch. In case of nucleic acid also, you know already nucleic acid, no? DNA and RNA. They are also having they are also not having any branch. They are not sweetish and do not exhibit any of the properties of aldehyde or ketone groups. Means they are not sweet. We have seen monosaccharides are sweet, disaccharides are sweet, but polysaccharides, some of the oligosaccharides, they are also sweet, but polysaccharides are not sweet, and they do not exhibit any reducing property. Aldehyde group or ketone group means they do not show any reducing property. They do not form usazone. That's why they do not. It is told that they do not produce any they do not exhibit any properties of aldehyde or ketone. So polysaccharide, there are multiple monosaccharides. These monosaccharides may be of single type. Say all may be glucose. Okay, all may be fructose. So when the monosaccharides are of same group, same type of monosaccharide, they are called homopolysaccharide. And when it is different, then it is called heteropolysaccharide. These heteropolysaccharide also called glycosaminoglycans or proteoglycans. Example of homopolysaccharide: starch, glycogen, dextrin. When starch, these are partially hydrolyzed, they form dextrin, acrodextrin, and this like that. Homopolysaccharide on hydrolysis, they yield only a single type of monosaccharide. They are named on the nature of the monosaccharide. Thus, when there is only glucose polymers, they are called glycans. When there is polymers of fructose, they are called fructogens. Examples are starch, dextrin, dextran, glycogen, cellulose, inulin, chitin. Chitin is the one which is functioning as structural form of homopolysaccharide. Starch, it is a reserve carbohydrate for plants. Example, potato, then sweet potato, any roots, tubers, they will contain starch. These are homopolymers composed of D-glucose unit held by alpha glycosidic bonds. Alpha glycosidic bond, here alpha 1,4 glycosidic bond by a glycosidic bond. It composed of two constituents, water soluble amylose 15 to 20% and water insoluble amylopectin 80 to 85%. Amylose is having less number of glucose molecules, say 1000 up to 1000, and amylopectin will have large number of glucose molecules, say more than 1000 to starting from 25,000. Okay. Amylopectin is a branch chain with 1,6 bond at branching point and 1,4 bond at every higher ends. So only in branching point it will be 1,6, other branches will be 1,4 alpha glycosidic bond. Starch is hydrolyzed by enzyme amylase to liberate dextrin and finally maltose and glucose. So maltose, why? Because it is 1,6 bond is there, 1,6 bond is there, that's why it cannot hydrolyze these two molecules of glucose, and others they are hydrolyzed into glucose. Here is the linkage. This is 1,4 linkage, this is 1,4 linkage, this is 1,6, and then 1,6, others are all 1,4. Here also all 1,4 it will be. Dextrins, these are partially hydrolyzed products of starch by acids or enzymes. Starch is sequentially hydrolyzed through different dextrins and finally to maltose and glucose. The various intermediates are soluble starch, amylodextrin, erythrodextrin and acrodextrin. These are the intermediate digestive products. All dextrins have 3 sugar groups and can show mild reducing property because these are the smaller polysaccharide and they have 3 sugar group. That's why they are weakly reducing in nature, but not as that of glucose or disaccharide. Glycogen, it is animal starch present in high concentration in the liver, followed by muscle and brain. Altogether, approximately 450 gram of glycogen is stored in the body, and they are responsible for immediate energy production. Whenever you fast, within few hours, you will need blood sugar to be established into normal range for providing energy. Now that glucose, after in the fasting state, after taking food, after 2 hours, your fasting state starts, and it will be till your next meal. So in between that meal, the blood sugar is maintained by the sugar derived from glycogen, by the glucose derived from glycogen. So the structure of glycogen is similar to that of the allopectin with more number of branches. Amylopectin with more number of branches. Cellulose, chief constituent of plant cell wall. Unbranched polymer of glucose linked by beta 1,4 glycosidic linkage. Beta 1,4 glycosidic linkage. In glycogen, what we found? It is alpha 1,4 linkage and alpha 1,6 linkage. Alpha 1,4 glycosidic bond cannot be hydrolyzed by animals the mammals, so they cannot digest cellulose. But animals, they can digest this cellulose because some of the organisms in their stomach, they synthesize these cellulase enzyme and they can digest cellulose. Since human lack enzyme cellulase, they cannot hydrolyze, cannot hydrolyze beta 1,4 glycosidic linkage. Cellulose cannot be digested and absorbed by the human. That's why it doesn't have any food value, unlike starch. Now, even though they do not have food value, it is very much helpful for the humans. Though not digested, has great importance in human nutrition. It is a major constituents of fiber, the non-digestible carbohydrate. The functions of fibers are non-digestible carbohydrates. Functions of dietary fibers include decrease the absorption of glucose and cholesterol from intestine. So, they reduce the possibility of the having diabetes mellitus. Okay. Now, if glucose is absorbed fast, there is a high rise of blood sugar. Okay. Now, that will increase possibility of diabetes mellitus. Again, in case of diabetic patient, to reduce sudden rise of blood sugar level, we advise them to take fiber type of food before their lunch or dinner. So, how we make available these fibers? We ask them to take salad before they take food. Okay. So, when they take food, it gets mixed up and the fibers, they interfere radially absorption of sugar and thereby reducing the possibility of high rise of blood sugar. So, increase bulk of stool, so there will be bowel movement regularly and aids intestinal motility, act as stool softener, prevent constipation. It reduces incidence of CVD, cerebrovascular disease, CVD, colon cancer, diabetes. Okay. CVD, cerebrovascular accident, because they reduce absorption of lipid. Harmful lipids, they reduce absorption of lipids and thereby preventing atherosclerosis. Atherosclerosis is one offending agent and it causes it causes ischemic heart disease. Okay. Myocardial infection, heart attack, what we say. Now, another homopolysaccharide is inulin. It is polymer of D-fructose. This is a fructosan linked together by beta 1,2 glycosidic linkage. It occurs in bulb of onion and garlic and also in dahlia. It is a low molecular weight polysaccharide, easily soluble in water. It is not utilized by the body, and this inulin, when we take, it is absorbed but not metabolized. It is absorbed but not metabolized. It is excreted in the urine as it is. So, the amount which we take is equal to the amount what we excrete in the urine. So, taking into account this property of inulin, the kidney function test is performed with this agent, with this molecule. So, it is used for assessing kidney function through measurement of glomerular filtration rate. Okay. So, we have taken certain amount of measured amount of inulin, and we will find out the rate of excretion of that inulin in urine. Okay. So that rate is glomerular equivalent to glomerular filtration rate. You will come to know the thing afterwards, but know this, it is done by this inulin. Chitin, it is a structural protein polysaccharide found in exoskeleton of some invertebrates like insects, crustaceans, then composed of N-acetyl D-glucosamine units held together by beta 1,4 glycosidic bond. Beta 1,4 glycosidic bond. Now coming to heteropolysaccharide, muco. This is also called mucopolysaccharides. Very important. You will be given short note on mucopolysaccharides or glycosaminoglycans. Same thing. Okay. So read this nicely. Heteroglycans made up of repeating units of sugar derivatives, namely amino sugars and uronic acid. Amino sugars and uronic acid, commonly known as glycosaminoglycans. Acetylated amino groups besides sulfate and carboxyl groups are generally present in their structure. Presence of sulfate carboxyl groups contributes to the acidity of the molecule, making them acid mucopolysaccharide. Now, another property of mucopolysaccharide is that it is very slippery. When you touch, you find that it is very slippery. It is because of the plenty of negative charges present in those molecules. So when there is plenty of negative charge, when you touch, and those charges, they tend to come together and there is repulsion. That's why it is slippery. Okay. Some of them found in the combination with protein to form mucoproteins or mucoid or proteoglycans. Mucoproteins may contain up to 95% carbohydrate and 5% protein. Mucopolysaccharides are essential component of tissue structure. Now, extracellular spaces in connective tissue, cartilage, skin, blood vessel, tendons consist of collagen and elastin fibers embedded in a matrix of ground substance. See, one type of tissue are there, in between the tissue one substance are there, that is called extracellular matrix, where the cells are suspended, and extracellular matrix that contains like this collagen, then elastin and ground substance. The ground substance is made up of this mucopolysaccharide. Important mucopolysaccharides are hyaluronic acid, chondroitin sulfate, heparin, dermatan sulfate, keratan sulfate, heparan sulfate. These are the composition. In hyaluronic acid composition, the disaccharide unit, repetition of the disaccharide unit. The disaccharide unit is here in case of hyaluronic acid, glucuronic acid and N-acetyl glucosamine. In glucuronic acid and N-acetyl glucosamine 4-sulfate. Heparin, it is glucuronate 2-sulfate and 2-sulfoglucosamine 6-sulfate. Dermatan sulfate, iduronic acid and N-acetyl glucosamine sulfate. Keratan sulfate, D-galactose, N-acetyl glucosamine sulfate. Remember, dermatan sulfate and keratan sulfate, they contain two different acids. Here it is L-iduronic acid and here it is galactose. Okay. Now these are the tissue distribution of different glycosaminoglycans and their function. This table I have quoted from Shaitanarayana, so you can get that, and in every book it is there. Okay, in Vasudevan also it is there. So here, just to show these structures are not required for you, just to see what is acetyl and acetyl glycosaminoglycan. And acetyl glucosamine. See, this is the glucose, This is in the second carbon there is amino group, and amino group is again acetylated. So the compound has become an acetyl glucosamine. It is nothing but the derivative of monosaccharide. Like that, others are also, they are having such type of, they are derived from the monosaccharide only. See here in this sulfoglucosamine 6-sulfate, it is having here one negative charge, here one negative charge. So like that, plenty of negative charges are there. Okay, so that's why these are very slippery. This is iduronic acid, nothing but the glucuronic acid only. In the fifth carbon, there is a inversion of the carboxyl group. See, this is glucuronic acid, the carboxyl group is upward, and here inversion of the carboxyl group, and that's why spatial arrangement, there is a difference in spatial arrangement of the carboxyl group. So it is ironic acid, and this is glucuronic acid. Hyaluronic acid. So there used to be some question. This time there was a question they wanted to ask. They wanted to know about the structure of structure of two glycosaminoglycans. So at least 2-3 you have to know what is that, so that you can write. Now hyaluronic acid, it is in synovial fluid, it is found in synovial fluid, ground substance, vitreous humor. Vitreous humor you know already, it is the jelly-like material behind the this one glands, posterior chamber of the eyeball. So forms gel around the ovum, ground substances in connective tissue, lubricant and shock absorbent in the joints. So in joints you will find some fluid which are very slippery, and their function is to lubricate the joint. Number one is lubricate the joint, and shock absorber also. Shock absorber, how it is, when you press, you put weight, the two bones, they try to come together, but inside there is fluid containing negative charge, and they will try to repulse each other. So there is a shock absorber function of that fluid. So that is nothing but the hyaluronic acid or glycosaminoglycans. The disaccharide, which is alternately there are alternate glucuronic acid and N-acetyl glucosamine. These are the disaccharide unit. So there is repetition of this disaccharide unit by beta 1,3. This is bound by beta 1,3 glycosidic bond, and disaccharide unit, each unit is bound by alpha 1,4, beta 1,4. Hyaluronidase is the enzyme which can break the beta 1,4 linkage of hyaluronic acid and other GAGs. Present in testis, seminal fluid, venoms of certain insects and snakes. You have seen that if some insects or snake bites, there is bleeding disorder, because the matrix, that intracellular extracellular matrix is hydrolyzed by hyaluronidase enzyme. That's why there is bleeding in after snake bite. It helps bacterial invasion of the tissue because it releases hyaluronidase enzyme, and there is easy dissolution of the extracellular matrix, and the bacteria can go inside. Hyaluronidase enzyme assigned important function for fertilization because it clears the gel around the ovum, facilitating better penetration of the sperm into the ovum. So it has a great role in fertilization. Now another is chondroitin sulfate, major constituents of various mammalian tissue, is found in bone, cartilage, tendons, heart valve, skin, cornea, etc. Structurally similar to hyaluronic acid, repeating disaccharides, glucuronic acid and D-galactosamine 4-sulfate. Dermatan sulfate is another, mostly found in skin, repeating. This difference between the Keratan sulfate and dermatan sulfate is that here instead of glucuronic acid, here hyaluronic acid there, as we have seen. Then Keratan sulfate, heterogeneous GAG with variable sulfate content. Disaccharide unit is galactosamine plus N-acetyl glucosamine 6-sulfate. Contains small amount of mannose, fructose, salicylic acid. Fructose, it is not fructose, F-U-C-O-S-E, fructose. This is another monosaccharide. Heparin, it is anticoagulant, present in blood, lungs, kidney, spleen, etc. Help releasing lipoprotein lipase, which clears turbidity of lipemic plasma. See, when you study spleen, you will find that there are sinusoids in the spleen and blood pools there. So wherever there is pooling of blood, there is a tendency of blood to get clotted. Okay? So that clotting should be prevented by something. It is the heparin that prevents clotting of that blood. It is the physiological component of the blood. Heparan sulfate. Heparan sulfate is a linear polysaccharide found in all animal tissue, found in basal lamina and other tissues. Repeating disaccharide units are glucuronic acid and N-acetyl glucosamine. Here the glycosidic bond is beta 1,4 glycosidic bond. Here this disaccharide unit is bound to another disaccharide unit of the same composition by alpha 1,4 lipid. So this can be hydrolyzed by the hyaluronidase. This is the diagrammatic representation of the proteoglycans. Here is the hyaluronic acid backbone. Here is the link protein. This is the core protein, and these black things, they are the chondroitin sulfate. So they look like this. Other heteropolysaccharides, agar and pectin. Agar is polymer of glucose, sulfate and glucose. It is found in seaweeds. These are dietary fibers only. Agarose is another material found from agar, derived from agar. This is useful in microbial culture media and electrophoresis. When you go to microbiology department, you will see that there is culture media preparation is there, where we put the organism and allow it to grow and see what are the antibiotics acting on that. Okay, so that culture media is prepared from agarose. And electrophoresis, one test is there. Electrophoresis, afterwards you will come to know. Electrophoresis, we use this agarose gel and we allow electric to run in that gel, and there is separation of different type of plasma proteins. Thereby we diagnose the abnormal plasma protein if present, and from that we diagnose the disease. Okay. Glycoproteins. Glycoproteins are proteins covalently bound to carbohydrate. Carbohydrate content is 1% to 90% by weight. Mucoproteins, when carbohydrate content is more than 9%. Examples are enzymes, hormones, transport proteins, structural proteins. These are one type of glycoproteins. Carbohydrate found in glycoproteins are mannose, galactose N-acetyl glucosamines, N-acetyl galactosamines, xylose, N-acetyl neuraminic acid. Glycophorins. Glycophorins are, these are the RBC membrane protein. What they do? They are glycosylated, 60% of them are glycosylated, 60% glycosylation is there, forming hydrophilic charge core, so that when they flow inside the vessel, they do not adhere to the other tissue. If there is adherence with other tissue, there will be clot formation. Okay? So that's why this glycophorins are very important in RBC surface membrane. Antifreeze glycoproteins in Antarctic fish, they have to live in about 2 degrees centigrade, minus 2 degrees centigrade. So there is possibility of freezing of the body fluid and lipid. But due to this antifreeze glycoprotein, it is not done. It prevents freezing or crystallization of water. Contains 50 repeating units of tripeptide alanine, threonine. Again, threonine is bound to beta glycosyl 1,3-N acetyl glycosamine, and this compound, it acts as anti-freezing compound. Blood group substance, you know, blood grouping, these are nothing but the glycoproteins. So in blood grouping, they play important, they are the important determinant for blood grouping. Okay. Now all these things you will know it, everything is not important for examination, but what are the things you have to know, so that in the next MBBS when you go, you know what are these things. So thank you for listening.